Welcome to Tuesday Talks. I'm Seema Kumar. I'm the CEO of Cure. Today, we'll speak with an incredible oncologist and genomic engineer turned entrepreneur whose company is working to transform treatments for metastatic triple-negative breast cancer and non-small cell lung cancer and many other cancers. Chidiebere Awah is the lead inventor and CEO of UTR Therapeutics. It's a preclinical biotech company based right here in New York City that emerged out of the Nucleate Activator program, which is a student-run accelerator. And it's designed to help scientists translate their findings into companies and products. UTR's breakthrough technology allows genomic engineers to better manipulate mRNA by allowing them to effectively target, overwrite, and degrade lines of RNA code called transcripts to potentially correct diseases, including difficult-to-treat cancers. Chidi's research and his discoveries have earned him multiple awards and grants, including the Novartis Institute of Biomedical Research Prize and the Dana-Farber Harvard Cancer Center Oral Award. Last year, Chidi and UTR Therapeutics were honored with the prestigious Exceed grant by Deerfield. Welcome to Tuesday Talks, Chidi.
Thank you, Seema. Thank you.
It's always great to see you here.
Thank you. It's my pleasure.
So before we begin and get into the details, I wanted to, get a little bit of your own personal background. Yeah. So you grew up in Nigeria, right? And
Yes.
you immigrated to the United States to do, advanced degree and further your work to combat cancer. How did that happen? And, how do you hope to bring your discoveries back to Nigeria to low and mid-level income countries?
Yeah. Thank you so much, Seema. It's really a very big pleasure for you to have me on here. I'm originally from the southeast of Nigeria. I grew up with my grandmother and my father in the southeast of Nigeria. And then I went to medical school. I had my MD degree at University of Nigeria, Enugu campus. And during the medical school, I really, during my internship, seeing patients, the pediatric patients and adult patients with cancer, I realized that the chemotherapies, the drugs that are available today are really not that effective. And so this spurred in me the urge, like I went to Cambridge University in the UK to then start to learn science because I discovered that there could be more to know and there could be more things to do. And then after that, I had my master's in Madrid, and then I was lucky to get into a PhD program with an incredible group at the Hanover Medical School in Germany. And it was in Germany that I really got molded into the kind of the scientist I am. I'm really grateful for my mentors there, Frauke Stanke, and my boss, Bukatumla. They were the people who equipped me and then gave me the skills, to pursue science without limit. And so after that, I came here for postdoc. Initially, I started working with brain tumor, and it was there that I discovered that mRNA and ribosome controls how tumors respond to therapy. And from there, I met my mentor, my long-term mentor, Kevin Struhl, with whom we laid the foundation of this work. And then with another mentor here in City University of New York, Olusegun Ogunwobi, we were able to have the proof of concept to develop this therapy, which I'm going to tell you more in details. And so if in wrapping up, we've been able to, take basic science, from that state medicine I came out from, I've been able to take this basic science back again, translated it into a product. And now we are very hopeful, right, and our drug is quite stable. So we are going to take it back again to Nigeria, to India, to all the middle and lower income countries, and people are very excited. This will be really the breakthrough they are looking for, where other therapies fail or are not even available.
So why is it so particularly suited for low and middle income countries?
So the idea here is that because our drug is in a DNA form, it's an RNA, but then it's encoded in a DNA form. And so therefore is very stable. You could ship it on 4 degrees across anywhere in the world. And this is the advantage we have against, like the vaccines and all that. So you could lyophilize it, you could put it in pocket, you could get on a plane, get on a train, and then take it to even where there are no light, it will still be very stable. So this is really the big thing apart from the cure itself that we provide, but that it will be available to almost everybody in the world.
That's fantastic, because then what you're saying is that you don't have the cold chain problems that typically, are really the barrier to, making this accessible to people worldwide. And because it probably doesn't require refrigeration, and it can be used in low resource settings and low infrastructure settings.
Exactly. This is nothing short of a miracle that we got the idea, from the RNA. The RNA message is in a DNA form, and so therefore you could lyophilize it, you could keep it at room temperature. So if you come into the lab, some of our drugs are there on the bench, and in the fridge is 4-degree fridges, so it could go anywhere in the world where there is no constant electricity. So this almost people in lower income and almost remote places of the world will be able to benefit from this.
That's just so fantastic. Now let's dive into the science a little bit, right? The science that is the basis of your company.
Yeah.
So tell me a little bit about how mRNA overriding works.
Okay, awesome. So this idea has been in the making for a very long time. So I was working on understanding, like drugs and even genes that control RNA methylation. And so I made quite a striking discovery in the sense that when I delete some of these genes, or when I use some drugs to treat this, the genes that control RNA methylation, I saw something very incredible, which is that you change some sequences on the 3' UTR of the genes. So the 3' UTR is the end of a gene. Mm-hmm. The gene has like a normal gene has like 3 parts. The 5' UTR at the beginning, the middle that is expressed is called the exon, and then the last part is called the 3' UTR. So most of this RNA methylation occurs on the 3' UTR, because 3' UTR has like a hub for integrating all the signals in a particular gene to make a full to complete transcript and to make a protein. And so therefore, what I saw was that if you then remove these enzymes that put the RNA methylation, or if you use the drug to treat it, you change some sequences actually, and these sequences turns off a gene or even brings that a gene on. So this was quite striking to me. And so once I saw this result, I ran back to Kevin. Kevin has been my long-term mentor, Kevin Struhl. He's a member of National Academy of Sciences, and he's a professor at the Harvard Medical School. Mm-hmm. And so I ran back to Kevin, and it turns out that Kevin has been working on mRNA stability in yeast for more than 20 years. I didn't know this. Mm-hmm. I know he knew because he's been very conversant with my work. And so I and Kevin, we then sat down and said, okay, are we able to, like these elements, right, that were changing on the 3' UTR to turn on a transcript and to turn off a transcript, can we see what the cancers have? And then when we look at the oncogenes, so oncogenes are the genes that cause cancer, it turns out that most of them have this mRNA stabilizing element. And so we came up with the idea, okay, so we are going to swap all those by motif engineering, engineer those mRNA stabilizing elements into a stable form. That's one part. And then we said, okay, if we then engineer it into an unstable form, we don't want it to go through DNA damage, which is unpredictable. We want it to go through nonsense-mediated decay. The nonsense-mediated decay is a process in the body. The body is a very versatile and unique machine in the sense that it does not tolerate transcripts that have problems. And so we then gave it a promoter that drives this destabilization through nonsense-mediated decay, meaning that if this transcript is destabilized, then it has to go through this very tightly controlled nonsense-mediated decay where that transcript is then degraded. And so we did this for let's say EP2 in many of the cancers that are undruggable or that are metastatic or do not respond to drug. And surprisingly, these cancers were like shrinking and collapsing. And we have even done it for some oncogenes like c-MYC or TED1 that are not undruggable because they don't have a binding pocket. And so this becomes like a brilliant idea where you could control any transcript without just that transcript and its interactome alone, without really having an effect on any other genetic regulatory network of the body. So that's the basis of the science. So it involved our engineering the stability element on the 3' UTR, but then asking the cell, now this has been destabilized, we want you to take it through the nonsense RNA quality control to degrade it through a very fidel pathway that errors are not tolerated.
That's fantastic.
Yeah.
And that's why, because you're working on the 3' UTR,
yeah.
that's why your company is called UTR Therapeutics,
Correct.
right?
Correct.
Yeah. So, I mean, to sort of reframe it in a lay language, what you're doing is you're taking this particular part of the oncogene.
Yes.
And then you are bioengineering it
Yes.
to make it unstable in
Correct.
some way,
Yes.
in a way that actually the body sees it as a error,
Yes.
and removes it.
Correct.
Right? And so you're actually deactivating.
Yeah.
So the oncogene is like a ticking time bomb
Yes.
inside your body.
Yes, exactly.
And what you're doing is you're basically taking those wires
Yes.
and removing, like unlocking it so that then it diffusing it.
Exactly. It is there, but it can't do any harm anybody.
It cannot do
any
harm
harm
anymore.
anybody to the
Yes.
person. So we have the message that is coming from, you got it exactly. You have the message that is coming from this oncogenic mRNA that is making tons of proteins and that is metastasizing and killing the patient. And so therefore we get it and then we destabilize it. We just diffuse it, and then the cells are no longer addicted to it, and we override that message. So we tell the body, you have this message, it's a problematic message, we're going to take care of it. Now it's going to be degraded, and it is no longer going to be used. So the body will now have to, that oncogenic message is no longer available, and then the cells are held back again, and they are going about their businesses.
And so, this is a basic discovery.
Yeah.
Have you actually tried to create therapeutics and test it on patients who have cancer?
Yes. So, like getting to patients is a process. So we have been able to validate this. First of all, we had the proof of concept right here in the City University of New York with Olusegun Ogunwobi, right? All the ideas were I and Kevin through, we conceived it, and then in New York, I and Shane, we started working on it. And we've proven it in all sorts of cancers, the triple-negative breast cancer, which as at last year, which we got an award for. And then we have also proven it in all sorts of non-small cell lung cancer and HER2 cancers. And now we have even gone beyond that. We've gone into more than 17 human cancers that are driven by c-Myc, and even more than 7 human cancers that all sorts of EGFR and drug-resistant breast cancer. And we've even drugged some other transition factors that are even undruggable, like TED1 and YAP1. There's no drug available for them. We've also, we've used it to create new therapies for it. And even a bigger problem is the Alzheimer and frontotemporal dementia. So it's estimated that about 1 billion people, are going to have Alzheimer or frontotemporal dementia at one point in time. By Thanksgiving last year, we got a target exactly for Alzheimer and for frontotemporal dementia, and we have validated this as well. And even for pediatric cancer. So it becomes because the technology is very basic, so you could apply it not just for cancer, but for metabolic diseases, for rare diseases, and for neurodegenerative disorders.
Wow, well, how groundbreaking, of a discovery this is. This is fantastic and phenomenal.
Thank you.
And so the thing is that you knew that other people might be very satisfied with just making this basic discovery.
Yeah.
But you didn't stop there, right?
Yes.
You decided that you wanted to start a biotech venture.
Yeah.
But as a full-time fellow, you needed the tools and guidance to get it off the ground.
Yeah.
So tell us how you kind of decided to make this transition using the nucleate activator program to move from basic discovery to starting a company.
Awesome. Thank you. So once we knew that this worked, right, and once we knew that we could use it to target all sorts of transcripts, whether they are cancer or disease cells from other areas, and so Sheun, who was my mentor here before, now he has moved to Michigan, and so Sheun saw the nucleate announcement, because it circulated across all the New York City institutions, and so he approached me with the idea. So I went to Nucleate, I registered, I signed up, and then when I signed up, so Nucleate is a very good program. It's run by students right here in New York, by all the universities in the New York City area here. So it comprises people coming with the technology, and then then these people are then matched up with students who have like MBA or finance ability, or people in Wall Street or whatever. And so then the Nucleate takes us through a coaching program that lasts for 4 or 5 months. And this coaching program tells you how a company runs, how to take a company from a product, how to pitch, how to talk to people, how to make your table. No longer the academic narrowed, process, but now how to actually bring a product into the market. And so it was there that I met Matt Megza, who is our lead business officer, and Riti Sanjraka, who is also one of our members. And so from there together, the 5 of us, we came together with Sheun and Kevin Struhl. And then one of our partners called Mena Ibulubu, he's also an MD. Mena has been working with me on the basic idea since 2019, the idea that led to the 3' UTR engineering. And so the 5 of us, we came and then we formed a company, and then we incorporated it. And so then we began to then take the product through the regulatory pathways.
That's great. I mean, it's not easy to start a company, right? What's been the biggest challenge and most surprising thing as you became from a researcher entering into like the commercial space?
So the most challenging, I think the first thing is that you have to have a solid science, right? If you have a solid science, that builds up your confidence. Like if you know that something works any day, anytime, anywhere, that is the first thing, right? You have to validate it across all sorts of things. And then the second part is getting to the business part, right? The business part, especially for scientists, they know already how to do the science. The second part is the financing, like the financing, like the seed funds. So we are very incredibly thankful for the SSEED award. This really helped us, it was a booster to us, right? And so we have not just done for one cancer or just one model. We have expanded this into for CMIC for 17 cancers. For the EP2, we've done it for almost 7 cancers. And so I think the funding is the limiting step for most scientists. And but I would say, first of all, have a solid science. The solid science is what will make you go to bed at night. You know that this is working. And then somewhere along the line, you have to hang in. The sun is going to shine. So you are going to get the funding.
If it's raining, don't give up hope.
Exactly.
Because the sun will come up again.
Exactly.
Okay.
As far as the science is solid, people will invest in you.
That's great.
Yeah.
So, of course, you need good science.
Yeah.
You need, seed funding.
Yes.
But then you also need the coaching, right?
Exactly.
From, because when you, as a researcher, you know everything there is to know about science.
Yeah.
But maybe you didn't learn how to start a company, right?
Exactly.
So how important was that for you?
So we are quite incredibly lucky because of Nucleate and the New York City ecosystem. I think we wouldn't have been able to do this elsewhere, right? Because here people are open, and then there's a lot of interaction. So we got a really lot of coaching from the Nucleate system. The Nucleate system, empowered us, brought external people, like speakers, the VCs, people always looking at your science from the other angle, right? Challenging the science and then making you to tighten up all the loose places in the narrative. And then once you receive this coaching, then you are very well prepared, to take this company. And then I also have a very strong supporting team. So I'm very thankful for that, and also for the New York City. So the New York City Economic Development Corporation has really helped startups like us. Like, they give us interns, which they are fully paid for. And our duty is to take these interns and train them. And then these interns are very talented. As you can imagine, the New York City is full of talent. So we have incredible people who are working for us. So you need to have all these 3 things coming together. One, you have to have that solid science. Then you have to have a coaching. It could be an accelerator. It could be like a group of student body. And then the next thing is that you have to have a support network, right? Matt, Riti, and all the rest of the people.
A great team.
Yes, a great team.
Yep.
And then the next is that you then have to have, like our city is really helping us. The mayor has done a lot by investing heavily in science, as well as the New York State governor.
That's great. That's wonderful.
Yeah.
And as you begin to translate your findings into a company and products, how are you making this, more attractive to institutions, to investors, and to pharma companies? I mean, what steps would you recommend to others who, like you, are sitting on top of like some great big science?
Exactly. Yeah, thank you so much for this. So we've started out right. So the institutional investors and the pharma companies, they first of all want to know that the science works, right? And this means that you have to if you have a proof of concept, which would be, let's say, in 1 or 2 models, so then you have to have a seed funding. The seed funding comes in very, very helpful, which is, we are incredibly thankful for the seed. The seed gave us the money, to be able to, then expand this to the breadth of it. So for example, last year while we are here, the triple negative breast cancer was the only model that we showed, right? So, but today we have done it for the CMIC that's undruggable. We've done it for all sorts of pancreatic cancer, all sorts of prostate cancer that are not responsive to therapy, all sorts of colorectal cancer, lung cancer, and even endometrial and ovarian cancer. So we've expanded this. So you need that seed funding to expand, right? And it is once you receive that seed funding, then this makes it attractive, like you could show the farmer that this is not just, like a fluke. It's something that is quite also applicable across board. Then the next thing is about the delivery, right? So most people have very good drugs, right? But these drugs are not well delivered. So for us, we're also quite incredibly lucky. Again, we have a second mentor, a partner. His name is Hiroshi Masui at the City University of New York. I work hand in glove with him. So Hiroshi has a delivery system of the iron oxide nano cages. So we use these iron oxide nano cages to package our drugs, and then we delivered it. And this is incredible. Even FDA recognizes that the tremendous therapeutic efficacy that we have is because of the delivery vehicle, which is the iron oxide nano cage. We're able to inhibit the tumors almost 80% across board, and even stop lung and liver metastasis.
Wow,
Yeah.
that's just phenomenal.
Yeah.
Well, so what's next? I mean, for you and for UTR?
Yeah, so for UTR, so our products are going now through the regulatory pathway. So for the first product, which is our CMIC, we submitted last summer we submitted a pre-IND to the FDA. And we had a very good interaction with the FDA. They asked us all sorts of questions. And so we have now gone back to the table, and we have now, they were asking us the dosing, the recruitment of patients. Who are we going to recruit? What will be the dosing schedule? What will be the GMP scale manufacturing of the iron oxide nano cage? And so we have all this now completely integrated. And by this summer, we are going to resubmit back that full package again to them. And then we also have a second product, which is even more as advanced as the CMIC, which is the EP2. These are the HER2-driven cancer. So they are mostly treated with trastuzumab. But within 5 years or so, these patients, they become resistant, or even with tyrosine kinase inhibitor for non-small cell lung cancer. And so we have also done the same for these EP2-driven cancers, about 7 of them, which non-small cell lung cancer that are EGFR T790 mutated, non-small cell lung cancer that have their EGFR and exon 19 deletion. We've also done this for esophageal cancers. Esophageal cancers surprisingly have the highest expression of EP2, and they are very responsive. And we've also done this for some colorectal cancer. So this is now being packaged, and hopefully by April, May, we are going to submit this package again. And so after that, then we would then start thinking of, our phase 1 recruitment. Yeah.
Well, good luck to you as you, embark on this journey.
Thank you so
and, with all the work that you do. It's just great. Thank you so much for being here with us, Chidi. It's wonderful. And to all of you, thank you for joining us today on LinkedIn. And don't forget to catch Tuesday Talks every week. See you again next week.